Today, I'm very happy to introduce Laura Martens, a colleague of mine. Some of you might still remember her as being the previous host of this Kipoi seminar series. Laura is a PhD student at Julien Gagneur lab at Technical University of Munich and Fabian Theis lab at Helmholtz. She is interested in sequence-based modeling and also single-cell world, and she's trying to bring all of them together in today's presentation about Scooby. Scooby is the first framework to model single-cell data from sequence. So from this, Laura, feel free to take over.
Thanks, Maria, for the introduction. Let me share my screen. Can you see it? Nice. Okay,
yes.
Wait, I'll just move. Okay, yeah, I'm super happy to present Scooby here at the Kipoi seminar, which I really enjoy. So yeah, I want to talk to you a bit about Scooby, which is a model that we recently developed in the Gagneur lab, me together with Johannes Hinger. And before I start going into the model, I just wanted to give you like the bigger question that we've been trying to answer, why we thought a bit about how to come up with this model. And basically, I mean, us among others, we're interested like how can we have such different or diverse cell states in our body, but also like every multicellular organism, even though each of the cell has the same identical genome. And I mean, of course, I won't tell you a lot new about this, but like it's really the precise and very well orchestrated readout of this genome and regulation of the gene expression that enables to have this one basically blueprint in every single cell, which is read out in super many different ways. And we really want to understand how this regulation works and how it is encoded in the genome. And then, of course, the second natural question that comes with this is at what point of this process can something go wrong, and when can variants, for example, interfere with this process, and at what stage would they interfere? So these are kind of the main questions that drive me and also our lab, and how we're trying to answer them, or one approach that we're trying to use to answer them are sequence-based machine learning models, and they all have in common that they use DNA sequence as input and try to predict something. But then once you have such a model, that can in principle be used for a variety of different tasks. So first of all, what I mentioned is like finding regulators, trying to pinpoint which parts of the sequence are actually relevant and regulatory. Then they could in principle be used to predict the effect of variants. And then thirdly, which I haven't really touched upon yet, is what also, like, for example, Steiner's lab, who's also in the call, showcased, is that you can use these models to actually design new elements with a promising or designed readout. And so one of the state-of-the-art models that does this, and this is kind of also how predecessor models worked, is Borzoi. And how this works is that it takes DNA sequence as input and it tries to predict some functional readout of this genome, which in this case is chromatin accessibility, transcription factor binding, CATAC-seq, which is a measure of gene expression, and then also Borzoi for the first time predicted RNA-seq. And the idea here is that you take this DNA sequence, one hot encoded as input, you pass it through a neural network, and you produce like richer sequence embedding, which should in principle already encode some details of the sequence, maybe if it contains transcription factor motifs and also other things, which can then be read out to produce the final prediction, which is the specific assay. And the idea here is that a model that has learned to go from sequence to these functional readouts, it should have learned some underlying logic of the DNA sequence and which parts are actually important to make this prediction. So they are super powerful and they can be used for interpretation. But the problem here is, and which is close to my heart, is that Borzoi is only trained on bulk data. So you can, of course, use it to interpret your sequence, but it will only be at the resolution of tissue and cell types. So what we were thinking was, how can we actually leverage this vast amount of single cell data that is out there that profile differentiation of so many different tissues and have this resolution really at single cells. So here on the right, I'm showing you a data set that we worked with a lot, which shows bone marrow differentiation going from hematopoietic stem cells into the three main lineages, the red blood cell lineage here at the top, then the monocyte lineage and the lymphoid lineage. And the question was, how can we use these sequence models to interpret this kind of data and maybe try to figure out what are actually the transcription factors that are important in each of these different cell states? Can we maybe find sequence determinants that would drive one or the other lineage? And then thirdly, what I mentioned, are there maybe variants that only affect a specific cell state and not other cell states? So when we were thinking about this model, one of the big technical problems were that these single cell data sets, they are just inherently super high dimensional. So you can imagine that you measure for each single cell, the gene expression for all the different genes. So how can we actually scale these models, which are by themselves already super computationally expensive, to this large single cell data sets? And can we maybe leverage cell embedding, so what people normally use to represent these cell states? So what we came up with is this model called Scooby. So this is kind of a reference to all the dog models that came before, like Borzoi, Bassett, Basenji, and then also the single cell reference. And what we did here, so I will explain the different parts. So it's like a two-part model. But the first part is that we leveraged the Borzoi model because we thought this model has seen so many, it has seen all the ENCODE, all the GTEx data. So it has seen a lot of very rich data, and it has probably learned a good basic representation of the sequence already and the motifs that are in there and the potential regulatory elements. So the first thing was that we used this base Borzoi model to create our sequence embedding. But what we add here, or what we added, is these data set specific parameters. In the simplest case, you can just think of them that they allow the model to adapt somehow to our new data set, because you could think of, first of all, single cell data is just it's different to bulk data. There's just different biases in there. But also biologically, you could think of that in our new data set, there might be motifs that should be a bit upweighted or more important for this data set. So in principle, these data set specific parameters should allow this base model to adapt the sequence embedding slightly to what it is trying to achieve. And then we added the cell-specific decoder. So in essence, you can think of it as just how you think a cell would read out the genome. You have a sequence embedding, which has all the transcription factor binding sites, for example, encoded in it, but each cell will use the sequence differently because it will only use certain transcription factor motifs, like one cell type, and the others will use others. So this is exactly what the cell-specific decoder is trying to do. So it uses a representation of our single cells, which in our case is like a lower. Those of you who are familiar with single cell data, is that the cell times gene expression data set, they're normally compressed into these latent or lower dimensional representations. embeddings. So we use these, which basically give the model some information on how similar cells are. So B cells will have similar embedding vectors, and then other cell types will have similar embedding vectors. And then the model will have to learn how to decode these embedding vectors and the sequence embedding to then predict the final coverage. So here we use this model to then predict, which is also different to other models. We really try to predict the coverage of RNA and of ATAC of single cells in this in the sequence window that we're looking at. I said to Maria that you can ask questions. So any questions to hear before I continue?
I do have one question. So this embedding vector of single cells, I guess I'm wondering like how well does it work if you have maybe like more discrete cell states, like if you had like let's say bulk cells and you had like T cells and B cells, would this embedding work well for that in terms of like telling similarity between cells, or is it more for like a single cell data set where you have this like large, where you have like all these different like continuous types of cell states?
No, I think we tried once with having like a mean bulk vector for every cell type, and this also somewhat worked. So
okay, thank you.
You're welcome. Okay, so this is the model, and then we evaluated, first of all, how well it actually predicts these profiles that we're training it with. And just because we've been asked this question a lot of times, so we really predict for single cells, but when we evaluate, we normally look at like the 100 nearest neighbors or pseudobulk just because it's a bit less sparse. So here, what I'm showing you to illustrate this is the observed RNA-seq profile for this locus here of the SLC25A37 gene. And it's a single cell and its 100 nearest neighbors. So each row here is really the profile of a single cell. And then when you aggregate it, you see this a bit nicer looking profile over the gene body, and you already see this RNA-seq bias, single cell RNA-seq bias that is usually centered around the 3' end of the gene. So when we then predict for a single cell, we see that it actually reflects quite nicely this expected profile of the 100 nearest neighbors. And the same for ATAC. So this is like, you can see this data is even sparser, but even here we see that Scooby can quite accurately predict this expected profile across the 100 nearest neighbors. So then when we try to evaluate this with using metrics, we faced a bit the problem, like what is actually the ground truth of this? Like what is the best profile you can predict? Because you can imagine that the actual measurements, they're actually like a very sparse estimate of the true accessibility. So we compared a few different things. The first thing so I'll just walk you through it step by step. So the first thing that we used kind of as the lower bound was how well would you predict a single profile? So this really sparse profile using the corresponding pseudobulk. So how well would the cell type profile correlate with the single cell profile, which is very low, as you can see here. And then we compared how well would a single cell profile correlate with the Scooby profile, which is better than this lower baseline. And then as a upper limit, we said, okay, how well would the sparse single cell profile correlate with the profile of its 100 nearest neighbors, which would kind of be the upper bound. So you see that Scooby basically has a higher resolution than a pseudobulk, but it's also not yet as good as using the 100 nearest neighbors. But then what we also compared, and which you already saw in the predictions, is that Scooby actually, the profile it predicts is like a bit of a, like the expected profile, or like it's less noisy than the single cell profile. So we also compared Scooby to the profile of the 100 nearest neighbors. And here we see like a very like high correlation both for the RNA-seq and also for the ATAC-seq. So the profiles that we predict, they kind of nicely correlate with the expected profile. And then we also went a bit more into gene expression prediction. So we could also benchmark with other models. So what we did is that we created a cell times gene expression matrix by summing basically the predicted coverage over the exons. And we did this for all cells. And to make this a bit less sparse, we also created basically cell type pseudobulk. So here, what we use is a cell type times gene expression count matrix. And then we evaluated both how well for one cell type we would predict the expression profile across genes, which is here on the left. So here we actually got really high correlations for the different cell types, so a mean correlation of 0.86 roughly. And then we also compared the harder task is how well do we distinguish cell types based on the expression of the genes, which is what we show here. So we also get a decent Pearson correlation, which is like in the range of what also Borzoi gets. But of course, this is a lot harder than the cross-gene prediction. And then we wanted to compare this to a similar model, which was also an inspiration for Scooby, actually, which is the seq-to-cells model, which also tries to predict gene expression counts. So I'll briefly just explain it to you and also the differences. So it also takes DNA sequence as input. It uses the predecessor of Borzoi, the informer model. So there's a shorter DNA sequence. And then it just takes the embedding of this informer sequence embedding at the transcription start site. And then it tries to basically decode this embedding for every single cell in the dataset to predict the gene expression counts. So first of all, you can see the difference is that it only takes this small sequence embedding, but then it only predicts gene expression counts. But then also because the model uses a weight for every single cell, so you can imagine if you try to train this model for large single cell data sets where you have like more than 100,000 cells or even 1 million cells, this becomes kind of infeasible because you really need to learn a weight for every single cell. And also when we evaluated the model compared it to Scooby, I mean, it still predicted well, but we saw that Scooby definitely significantly outperformed the seq-to-cells model on both the across-gene correlation and on the across-cells correlation. So yeah, now I've hopefully convinced you that the Scooby predicts somewhat reasonable predictions. I want to show you some use cases that we thought of with Scooby. So first of all, what I mentioned, how well can we predict transcription factor activities across cell types? So what we came up with to basically probe this was this, what we call in silico motif mutation. So what we did is that we took the DNA sequence around a gene, and we scanned this sequence for instances of transcription factor motifs that we got from the Hokomoko database. And then we basically deleted or shuffled, no, we actually randomly replaced every instance we could find of this motif by a random substitution. And then we recorded the difference of both gene expression and accessibility of this unchanged sequence compared to the sequence that didn't have the motif. And what you can think of is that if a transcription factor motif is important for a cell, then this difference here should be bigger than if a transcription factor is not important for a cell. So here I'm showing you an example of how this would look like. So this is the early B cell factor 1 and its effect on gene expression for every single cell in the dataset. And we see that the model predicts the highest effect here in this lower population, which is in fact the B cells. So this makes sense. But we also evaluated this more systematically. So what we, again, there's not really a ground truth for transcription factor activity. So what we used as a proxy is just how well we correlate with transcription factor expression. And we compared this both to ChromeVar, which I'm showing you here, and then also scBASID. But I also want to, that's not the point I want to make here, but what we saw is that we better correlate with gene expression compared to both ChromeVar and scBASID, which is better is here in this gray triangle. But also interestingly, we saw that we also get kind of decent predictions on a model. So this left model here was trained with both single-cell RNA-seq and single-cell ATAC-seq. And all the other methods that try to infer transcription factor activity, they usually rely on single-cell ATAC-seq. So we also trained a model only on the RNA data. And we saw that even there we get kind of a reasonable correlation with the transcription factor expression. So what we wanted to show here is that Scooby can also be used to infer transcription factor activity, even if you don't have accessibility data available. And then I mentioned that we can measure the effect on transcription factors, both on the RNA and also on the accessibility. So what we looked here is, again, this differentiation from hematopoietic stem cells to the normoblast cells. And one important transcription factor for this differentiation is scATAC-1. So we looked at the effect of the GATA1 deletion, both on the accessibility and on the RNA-seq. And we basically saw that Scooby predicted this accessibility expression leg. So GATA1 deletion had first an effect on accessibility and then on expression, which also biologically made sense to us because GATA1 is also a pioneer factor. So it first comes and opens the chromatin and then it binds to start the expression. Yeah, so that was nice to see. And then, second of all, we looked into if we can maybe propose sequence determinants. So in this case, we saw that we can correctly, or that we can predict the transcription factor activity along a trajectory, along a differentiation trajectory. And we also wanted to see if we can maybe propose sequence determinants that could drive cell fate transitions. So we looked into this really nice data set, in my opinion, which is hard organoids. So our collaborators, they generated hard organoids and profiled them with single-cell RNA-seq and single-cell ATAC-seq at different time points. So you really nicely see this differentiation of early cell states to then later cardiomyocytes and epicardial cell states. And the population they were really interested in were this JCF population here, because they have the potential to both differentiate into epicardial cells and also in cardiomyocyte cells. And in this publication back then, they basically identified two subpopulations in this JCF cells that were more prone to differentiate to epicardial cells and cardiomyocyte cells using cell rank. And what we did, just to see if we find transcription factors that correlate basically with these two transition probabilities, is that we used our predicted transcription factor activities or motive scores and correlated them with these transition probabilities. So here on the left, you see transcription factors that are more have a higher correlation with the epicardial transition probability. On the right, transcription factors that have a higher probability with the cardiomyocyte transition probability. And we saw some candidates that really made sense to them, so like FOS and GATA4, for example, here. And what was also really nice when we actually looked into the predictions was that we could see that Scooby can really, even in this like one cell type, predict differences basically in transcription factor activity between different cell types. So here in the top, I'm showing you FOS, which had the higher probability for higher correlation with epicardial cells, and then GATA for cardiomyocytes. Yes, so the last one is basically, I guess, the problem that a lot of people try to solve. So I want to present you a bit on what we try to do here. So basically, how well are we at predicting variant effects, and can we also maybe predict variant effects that have like cell type specific impact? So yeah, the question here is basically you can imagine a variant that affects a transcription factor, or that is only relevant in one cell type and not in the other. So it would also only have an effect in cell type 1 and not in cell type 2. So what we did here to benchmark this is we used two different data sets. One is the 1K1K data set, which is a very huge single cell RNA-seq data set, which has more than 1 million cells of roughly 1,000 donors, and what they called EQTLs for 14 different cell types. And then we also compared it to GTEx, which is a bulk resource, which, for example, also Borzoi benchmarked with. So we trained Scooby. So this is already was also cool to see that we actually could train Scooby on a data set that had 1.3 million cells. And then we compared how well our predicted variant effect correlated with the observed variant effect. So what we did is that we, or how we defined our variant effect is that we took the difference in expression, in predicted expression for the reference sequence compared to the sequence with the variant. And then we correlated this predicted variant effect with the observed effect. And here in blue, I'm showing you, so here we are comparing Scooby against Borzoi, and in blue, I'm showing you the bulk data set. And it was nice to see that we are slightly worse than Borzoi, but we don't really trade a lot of resolution for performance here. So we still predict these variance effects somewhat well. And then on the 1K data sets, so what we did for Borzoi to have cell type specific predictions was that we tried to find bulk tracks that mostly match the cell types, or that best match the cell types that we were trying to predict for. So each dot here is one cell type, and we saw that Scooby outperforms Borzoi on all of these different cell types. So this all to say that Scooby can predict at least better than Borzoi, variant effects in different cell types. But then we also, and this is maybe what some of you will be very interested in, we also looked a bit more into how well are we really at these variance effect predictions. And what we saw, and this is not only true for Scooby, but also for Informa and Borzoi and seq cells, is that for a lot of variants that actually have a strong observed effect, the sequence-based models don't really predict an effect. So these are all the variants that fall here into this red ribbon. But the interesting bit maybe is that for the variants where Scooby actually predicts a high effect, so these are the ones that fall outside of this red region, we have actually a much higher Spearman correlation. So we get a Spearman correlation of 0.8 roughly. And also interestingly is that most of the variants that have these higher predictions, their sign is correct. So we get like more than 90% of the variants actually have the correct predicted sign. And we observe the same for Borzoi also. And then, that's here Borzoi. And then we also looked into how well do we predict variants with larger distance, like distal variants. So here, again, I'm distinguishing between the variants that like all the variants, and then variants that where we actually had a higher predicted effect, which are the red ones. So for all the variants, we see this effect that a lot of these sequence-based model papers have reported, basically that we are pretty good at predicting the variant effects of close variants, but the sooner, like the further the variants are away, the worse we basically get. But also what we found interesting is that for the ones where we actually predict a high effect there, we don't really see this decaying effect. So yeah, the take-home message here is that for variants where we predict a stronger effect, you can normally trust the sign concordance, and for further ones, then not so much. Okay. I don't know if there was a question. And then lastly, I wanted to we looked a bit more closely into can we find variants? Can we find variants that basically have differential effects between cell types, or that only affect a specific cell type? And the idea here was that in principle, we could also take the bulk EQTLs that have already been called and use single cell data to basically deconvolve their effects and pinpoint which cell types they might actually affect. And in the paper, we also evaluated this more systematically, but I want to show you here one case study or one example where we looked at the test gene, and basically this Scooby for this gene predicted a high negative effect for the monocytes, and not like basically no effect for the erythrocytes. And also when we looked at the actual predictions, so here I'm showing you the profile, the difference between the alternative and the reference predictions. So we see that for the RNA expression, there's lower expression for the alternative, whereas for the erythroblast cell lines, there's basically no difference. And then also for the accessibility, so here in this light gray, I'm showing this is the location of the variant. We saw that the predicted accessibility at this location is reduced. So the accessibility at the variant location basically closes. And again, for the erythroblast cells, we didn't see this difference. And then we basically looked into the model and tried to interpret why this is the case. So here I'm showing you the importance of the underlying sequence that the model predicts, and in the reference sequence for the monocytes, it basically highlights this motif here in the middle, which is the SB1 motif. And you can see that the variant that is introduced is here, this G to A, which destroys the SB1 motif. So then the model doesn't highlight this part as important anymore. Whereas in the erythroblast, it doesn't highlight this sequence in the first place. So there's also no effect by disrupting the motif. And then when we looked at the expression of the SB1, we also we saw that this transcription factor is indeed only expressed in the monocytes, not in the erythroblast. So with this, I hope I could introduce you to Scooby and what the idea behind it is and how it works, and how it can basically scale to very large numbers of cells and by leveraging Borzoi. That we can accurately predict the accessibility and expression profiles, and also the gene expression counts. And I showed you some examples of how we thought we can use it to get cell state-specific sequence determinants and cell type-specific EQTLs. And with this, I would like to thank my co-first author, Johannes, who, yeah, we worked a lot on this together. And then all my co-authors, and then, of course, the whole Gagneur lab. And then this is the link to our preprint. And with this, thanks a lot for listening.